Obviously, as you mentioned, the challenge here is with diagnosis, and of course, especially in the acute time frame. And you've done some extensive research in this area. Can you walk listeners through some of your earlier work and what you guys were trying to uncover, and how these acute biomarkers might serve the athletes, the sport, and the medical staff to be able to really get the best outcomes for everyone?
Yeah, absolutely. So in terms of the what we've talked about, I think we've made it sound pretty easy to pick up concussion, to diagnose it.
Nothing to it.
Yeah, it's just so simple, which is kind of doing a bit of a disservice. Like sometimes, it is straightforward because someone's been knocked out, or they're telling you, like, I feel absolutely dreadful, and I don't know where I am. But in quite a lot of cases, the symptoms can be a bit. Yeah, the symptoms can be vague, and they can take time to develop, and people aren't hiding any symptoms. They're just not sure, and fair enough. So there's a big interest in the whole of the kind of concussion field at looking for an objective biomarker of concussion. So maybe the player doesn't want to come off, or maybe the player doesn't recognize that they've got symptoms, or maybe the player genuinely does not have symptoms yet, but they're going to get them in 24 hours. If you had something that reduces the subjectivity and just makes it black and white, then that would be tremendous and a big improvement in terms of player safety. Not just in sport, but also
in life as well, right?
Life, like people hit their head and present to emergency departments, or sometimes if they have an unwitnessed injury, they don't present to anyone, but they just feel rotten for a few days and they don't know what to do about it. So what our team was looking to do was to see whether we could work with the World Rugby head injury assessment process as it's applied in elite rugby in England, and collect saliva samples from players going through that process to see if we could identify salivary biomarkers of concussion. The reasons for looking in saliva are that it's a really practical and accessible fluid for settings like dirty settings, like field-based and that kind of thing. So you don't need to have a phlebotomist or sharps equipment and stuff like that. And the kind of background to it, that wasn't the only reason for looking in saliva. What we were looking for were markers called microRNAs, which may or may not be familiar to your listeners.
Yeah, go ahead and give us define it, and it'd be great.
So microRNAs are, as the name suggests, really small bits of RNA, and they're not a new class of signaling molecule, but the technology to look at them and study them on a big scale hasn't really been around for that long. So at the moment, we're seeing a real boom in people looking at them because basically they can, and they haven't been able to do before. And what they do in the body is they regulate the expression of proteins. So they will bind onto the bit of the cell that's putting the protein together, and most often they'll silence that process. So they'll stop the production of a protein. So it's a little bit like, our DNA contains the recipe, the instructions for producing proteins that we need for function, and then microRNAs fine-tune that process. So little subtle changes that we need to kind of maintain normal function. And because there are so many of them, there are thousands and thousands of different microRNAs, and each one can have a lot of different functions, and each different protein can be affected by a lot of different microRNAs. It's a little bit of a speculative exercise to try and see, can we see patterns that
what's doing what, right, in terms of the influence. And
exactly, can we see patterns that will signify injury? So what the team that I worked with at the University of Birmingham looked at initially was to see whether microRNAs in blood could distinguish different severities of traumatic brain injury in road traffic collision victims. And that work demonstrated that, yes, there are patterns of markers which are different in people with more severe brain injury and less severe brain injury. But obviously, as we discussed, blood isn't the perfect fluid. So then we tried to look and see in saliva in patients coming out to our concussion clinic, could we see differences in the concussed ones and in some healthy controls in their salivary microRNAs. And that was successful also in showing, yes, there are different patterns of expression in these people. So that was the kind of reasoning behind doing a big study involving Premiership Rugby and Championship Rugby in England and the RFU, to say, okay, let's collect a lot of saliva from a lot of players going through the head injury assessment process and see if we can pick up specific changes. I'm conscious that I may not be making any sense.
No, this is great. I think everyone's going to be on the same page, and it is, again, in sport, whether it's ice hockey, whether it's American football, we see obviously in soccer, what the rest of the world calls football. The rates are seemingly on the rise, and in the general population as well. And so obviously this point of care testing where we, for medical professionals like yourself and performance staff to be able to get a real time assessment of where things are headed. I mean, that's invaluable. So in terms of, the players, how many players did you end up collecting, and walk us through what you guys uncovered?
Yeah. So we had a really terrific response from the teams and from the players that we worked with, and as well as the team at the University of Birmingham who worked on this, and the RFU. I have to say, I can't thank enough all of the teams and players in England who helped us with this, because they were absolutely fantastic, and we were able to recruit about just over 1,000 players to the study.
Incredible.
And then from that, we got about 150 concussion samples from about 150 concussions over the course of the 2 years the 3 years rather that we were running it. The coronavirus kind of brought things to a
bit of a halt,
premature halt.
Worldwide pandemic threw a little wrench in there.
Yeah, that's it. And we had an uncomfortable moment where we said, coronavirus is an RNA virus, and we're doing everything we can to protect the RNA. So maybe we might want to put these in the fridge for a little bit and come back to them later.
Yeah, exactly. Yeah.
But so how we set the study out was we collected samples at baseline. We collected samples from players who were our control players who had just played a game. So they've played elite rugby. We know that they've had a degree of subconcussive, trauma in what they've been doing. And we collected samples from control players who had musculoskeletal injuries. So they had, a sprain or a muscle tear or a fracture that caused them to have to come out of the game. And so by looking at differences between the samples we got from athletes with concussion and those control samples, we got a bit of a proof of concept that, yes, there are differences in salivary microRNA expression that you're seeing because someone has concussion. It's not just because they've been through, really hard physical work. It's not just because they've had some kind of trauma to their body. It does seem to be a different pattern when it's brain trauma. And so that was really, encouraging.
Pretty exciting.
That was a really exciting finding. But then we were able to follow that up and to look at the group of people who went through the head injury assessment process and concussion was ruled out, and the group of people who went through the head injury process and were diagnosed with concussion, which is probably like the most relevant thing you want to know, because those are the people you're actually going to use your test in.
Yeah, precisely.
Sometimes we have these sorts of conversations where we look over studies that have been done in the field, and often the comparison is to people who have, orthopedic injuries, and that's really useful. But you think, I'd never have a problem working out if someone's leg pain.
Yeah, real time scenarios. It's like, this is not an issue with
exactly. Like I don't need to, if he's got leg pain, I don't need to
I don't
rule
need a
out.
special test.
So yeah, and so in that comparison between, you know, the concussed group and the concussion ruled out group, again, it demonstrated that there were differences in expression between those two groups, and that you could construct a model to kind of separate them out. So we're hoping that in the next few months we'll have the final version of the paper accepted and ready to go into print.